Good morning and welcome back. So, if I sound like I'm talking quietly, it's very early in the morning. Don't want to wake up the dog above all. So, yesterday I talked about my real taste for molecules as documents of evolutionary history. Of course, it's like a front and center as one of the most important parts of modern biology, so I'm not making any sort of claim of creativity on my part. I'm just saying that I'm definitely a fan. And now what I want to do is I want to use hemoglobin as a case study in order to illustrate how the use of molecules to document of evolutionary history has played out. And the the example that I guess I'm going to start with, and the reason I call it genomics without genomes, is this classic paper from Zuckerkandl, Jones, and Pauling. And the reason I like it so much is that I really believe in the importance of indirect science. And what I mean by that is that there's so many things that we know in the scientific sense, and we truly do know them, and yet we haven't necessarily, or at least when the original discovery was made, we didn't observe it directly. We knew about atoms before somebody had manipulated one with a scanning tunneling microscope, on a surface, or we, we knew about the disposition of genes on the chromosomes of the Drosophila chromosomes well before anybody even knew that DNA was the genetic material. That was the work, for example, of Sturtevant and Thomas Hunt Morgan, where what Sturtevant was able to do was to take the recombination frequencies and look at pairs of traits and to ask the question of whether or not they segregated together during recombination, and you could see the closer two genes are on the chromosome, the more likely it is that when you cut the chromosome somewhere in order to do recombination, that those two genes will segregate together. And so, based on frequency of joint segregation, Sturtevant was able to map out the physical distance between genes. And what I want to say is, that took place in the 1910s era, long before it was established that DNA is the genetic material, first um, by McLeod, McCarthy, and company, and then after that Hershey and Chase, and so that was 30 years later. Oswald Avery was the last of those people. So we nowadays you can sequence Drosophila and you can find out where these different genes are, and what you'll find is that what was established earlier was already true, correct. So here, this is before we knew how to do sequencing, at least at the scale it was needed for this comparison. And what Linus Pauling and Zuckerkandl and Jones did was that they digested the hemoglobin, meaning they cut it with trypsin. It got cut at certain places, and then they ran the protein on a two-dimensional gel. And by comparing the gels from different organisms, you effectively are comparing the similarity of the protein. So I'm going to come back to that in a little bit. So hemoglobin. So hemoglobin is a poster child of modern science. And what I mean by that is that it has over and over again been a gift that keeps giving. In many ways, like the hydrogen atom has been in the subject of physics. So, Max Perutz founded, in many ways, was one of the key founders of modern structural biology. And he spent a good part of his career focusing on hemoglobin and its structure, which in turn led to deep insights into the super powerful notion of allostery, which we'll talk about in coming weeks. And it's been a really, really central player in the statistical mechanics of binding and how we think about cooperativity, especially. So it's a really important case study. And here I show you the lungs and a close-up showing a blood capillary with red blood cells inside of it. And inside of those red blood cells are hemoglobin molecules. If I remember, it's something like 300 billion, 300 billion hemoglobin molecules per red blood cell. And the hemoglobin it binds oxygen, as you probably already know. And so the structure, as you see here, it involves these four heme groups. The oxygen binds where you see the iron on the right-hand side. And so anyway, it's a key molecule in the physiology of organisms, and it's something we can say a lot about from the standpoint of statistical physics. I've always just been enamored of the fact that Stokes, George Gabriel Stokes, who was a very unusual character. If I remember his story, he was I guess the thing I was going to say was that Stokes is known to many of us if we practice physics through theorems that we encounter in the context of vector calculus. But he also became fascinated by the spectrum of hemoglobin, and specifically of how the spectrum is altered by the binding of oxygen. And so, I think that's a really cool topic. The notion that you have this molecule, and depending upon whether or not it's liganded with its target molecule, its spectrum will be different. And we might talk about that a little bit. So I had already mentioned the fact that its binding curve, the binding curve of hemoglobin, is really central to our understanding not only of physiology but of how molecules work. And what you see on the x-axis here is the partial pressure of the oxygen. What you see on the y-axis is degree of saturation. So when we're at 100%, what that means is that all four sites on the hemoglobin are bound with oxygen. What we see when it's zero is that means that none of the sites are occupied. One of the things that I want to call to your attention is the sigmoidal character of this curve. What I mean by sigmoidal is that there's a change in the curvature. It starts out going up like this, and then it turns over. So that means if I evaluate the second derivative changes sign as a function of concentration of oxygen. Well, so what? Well, if you do the most naive model of binding, what you will find is that it does not suffer a change of curvature. It has only a negative curvature. And that's a so-called Langmuir binding curve. And so Christian Bohr, who was the father of Niels and Henrik Bohr, I think was his, I don't remember Niels Bohr's brother's name, but he was a famous mathematician. At any rate, he discovered the effect that you see here, which is increasing CO2, the binding curves are shifted, which is, a very interesting biological fact, and tells you something else, which is that, yet again, this is a kind of molecule that has more than one partner, which is I think of that as being a very biological fact. If I consider one of the most important enzymes that all of us learn about, which is phosphofructokinase, this is relevant for glycolysis. That enzyme has multiple inhibitors and multiple activators. So it's just to point out that these are all concepts that had to be learned that are now quite overarching. And I, as somebody who likes to think of the action of biological phenomena in a mathematical way, very much appreciate the challenges that are offered by a molecule who has multiple ligands that alter its destiny in different ways. So, okay, with all that as background, as to what hemoglobin does, this is 5 separate panels from a figure from the paper of Zucker, Candle, Jones, and Pauling. And I'm not going to go into the details of it, because really the only thing I wanted to show you is I wanted you to, in a certain sense, say, wow, if you're, in your 20s or late teens and a student and listening to this, I'm venturing the guess that this idea of genomics without genomes hadn't really been a part of your repertoire. And I think it's very important to know, if for no other reason than because, these scientists wanted answers. They couldn't say, I'm going to blast it, go to Protein Data Bank or something like that. It didn't exist. They wanted the answer. So they had this incredible ingenuity to imagine that, well, if I cut up the protein and I run it on a gel, I might be able to distinguish the nature of the spots on the gel, and from that I could infer something about the evolutionary history of this molecule. This is a more modern view. So this is still not exactly the way necessarily that one would think about it today, which would probably be at the level of the DNA. But here, what you see is the protein amino acid sequence for the 142-ish amino acids in one subunit of hemoglobin. And what I want you to note is for example, if you compare the two rows called human and chimp, you'll see that they have no differences. There's 0 amino acid differences between the human and the chimp hemoglobin. The gorilla has some differences. I can't remember how many, but if you go to cow, you'll see even more. For example, here 1, 2, 3, 4, 5 at the fifth, already at the fifth spot. You see that there's a difference and so on. And so this is very interesting because there's an idea behind the scenes of this notion of a molecule and its sequence as a ticking clock. I am not going to enter into the fine points of this. There's really, really interesting literature where people have thought very hard and debated and discussed and tried to analyze and calibrate these various clocks. I'm going to, for the purposes of this particular vignette, offer it as a given, but cognizant of the fact that there are some points of contention, some points where we can have doubts and so on. That's fine. So what you're supposed to see here is that for a given molecule, the nucleotide substitution rate depends upon the time, and the very fact that I'm giving this vignette, even though now I'm quite a fan of it and may well continue it indefinitely. At this point I'm teaching in the classroom. I've already actually done this vignette, but I find the quantal nature of doing vignettes really satisfying for me, and in terms of the way that I like to structure my course, which is, and how I learn, which is I want to have elemental ideas. So this particular vignette has one idea, which is how would you do genomics without genomes, with the specific case study of hemoglobin in mind. Okay, so what does that have to do with anything? The reason I'm doing these vignettes is because we're living in a global pandemic. Well, yeah, what about it? The global pandemic is due to a virus which is known as SARS-CoV-2. It's very interesting, super interesting virus. It has a genome of roughly 30,000 nucleotides. And one of the most interesting features of this particular virus is that it has a proofreading, meaning that it does error correction. Nevertheless, it's changing over time. We're now really on the border of 2 years from when I personally and many others went into a strict lockdown. And one of the things that you can go do is you the listener can go look at the website called NextStrain. There's a reason for this whole digression. If you go to NextStrain, you can actually do a plot like this one that shows you the accumulation of substitutions in the genome over time, and what you'll see is that it's got this kind of a look to it. So even though the one I'm showing you here is a divergence time of 50 to 100 million years, that one's got a divergence time of months. Right now we're annoyed by the Omicron variant, and it follows on the heels of the Delta variant, and you can just look at what happened. Betty Korber, I think, told us about the first of those variants. Anyway, I'm not going to pass through these the next few slides with much care. I'm just going to sort of show them as tourism and let you, as the listener, go and do your own explorations. But the thing I want to say is that, in the previous vignette, I talked about phylogenetic trees, and here I'm just noting once again, a reminder of where they come from, which is the more distinctions there are between two different organisms, the the farther into the past lives their common ancestor. And so, using these kinds of ideas, you can take the organisms that I showed you, their hemoglobins for, and you can attempt to build a phylogenetic tree. And what comes out of that for hemoglobin is shown here. So this is a tree that's based on the hemoglobin sequence. You can see that at the top we have human and chimp, and then gorilla, and then the next one in the group is the cow. And as we go farther back in terms of phylogenetic distance, we're going farther and back in time to the most recent common ancestor. So this notion of the molecular clock, hemoglobin is one of the exemplars of this, and there's something about the y-axis here that I find odd. I think I've never even noticed that before, so with apologies. What you see here is 3 different molecules, cytochrome C and hemoglobin, and then the fibrinopeptides, which, if I remember correctly, are related to blood clotting. And what you see is the relative stability of these different gene products over time. So I think this is the main things that I wanted to say about this first case study of hemoglobin. I really just hoped to give you a taste for this kind of idea, that we can use sequence as a window onto evolutionary relatedness. I wanted you to be deeply impressed with this, which is getting sequence relatedness, but without actually doing any sequencing. So what we'll do in the next vignette is we'll talk about Alan Wilson, one of my great heroes in science writ large. And but for now, this vignette was about hemoglobin.